Hello everyone, welcome back. My name is Dr. Vijay Bhatia and today I'm going to talk about mechanisms of action in the brain by the alcohol. If you haven't seen my other videos, please see them to see the structure of alcohol and what are the toxic effects of alcohol dependence, symptoms and symptoms of withdrawal. In this one, I'm going to focus mainly on the effects of alcohol in the brain on the neurons. Alcohol actions in the brain. Alcohol enhances inhibition of GABA synapses. Alcohol reduces excitation at glutamate synapses. We will talk these in more detail. GABA means gamma-aminobutyric acid. This is the main neurotransmitter in the brain which slows down the stimulus, the impulses. So alcohol helps to enhance and thereby slowing down the brain firing of the brain neurons. Whereas glutamate, it's an excitatory neurotransmitter. So alcohol reduces the excitation, therefore helping the brain to slow down in firing and communicating with other neurons. Alcohol actions at GABA, that means gamma-aminobutyric acid synapses, enhance GABA release via blocking presynaptic GABA beta receptors. So what happens is there is a GABA receptor and it is in the presynaptic neuron. So alcohol actions at GABA synapse, this is a GABA synapse, and there are this presynaptic GABA beta or GABA B receptors and directly stimulate postsynaptic, and in the postsynaptic membrane there is the GABA-A receptors, especially those of the delta subtype that are responsive to neurosteroid modulation but not to benzodiazepine modulation, either via direct actions or releasing neurosteroids. What I'm trying to explain is this is a GABA receptor. What happens when the GABA receptor is open, the chloride ions in the extracellular space outside the cell will enter the inside the cell when it is open. And what happens normally inside the cell there is negative. If I try to put more chloride inside, it will become more negative. If it becomes more negative, it means it is hyperpolarized, so it will slow down its firing. Now, how does chloride get into this? Is through the GABA receptor. Receptors are like gates in the cell membrane of the nerve cells. Normally, this GABA receptor is made up of these 2 alpha, 2 beta, and 1 gamma strand. When GABA itself, as a neurotransmitter, which is released from the presynaptic neuron, attaches here, it will open this and chloride will come into this. There is another allosteric site where benzodiazepine will sit. If somebody has taken benzodiazepine, they will sit here and it will cause positive modulation, and the chloride will open more and more chloride will go in, therefore making it more hyperpolarized. And of course you will need the GABA neurotransmitter there. But here at this side, if the benzodiazepine will sit here, but this side can be also occupied by some neurosteroids inside the brain. And there is another site here, modulatory site, which is for barbiturates. But normally we don't use barbiturates very much but they used to sit here and open it and help it to facilitate the GABA receptors. And once the GABA is open, chloride will come here and it will slow down the firing of the cell because of the hyperpolarity, more negative inside. What happens? This is same GABA receptor. GABA receptor has another site where alcohol molecule will attach, and it will have the similar effect as positive modulation, where chloride will come into here and enter into the nerve cell, making it hyperpolarized, therefore less active and less impulse flowing to the other nerve cells. So it is playing the role as if it is a benzodiazepine and as if it is sitting here on the side here to help facilitate GABA receptor. Again, these are 2 alpha, 2 beta, and gamma delta receptor, and this ethanol has a site on the delta receptor of the GABA receptor in general. Alcohol acts at presynaptic metabotropic glutamate receptors. So now we are talking that the previous slide we were talking about the GABA receptors, but now we are talking about glutamate receptor. Glutamate receptors, again, this is a presynaptic and that is a postsynaptic membrane. This is a synapse, this is a presynaptic neuron, postsynaptic neuron. Now this glutamate is excitatory. Alcohol acts at presynaptic metabotropic glutamate receptors. So here they are glutamate receptors in the presynaptic membrane and in the postsynaptic membrane. And presynaptic voltage sensitive calcium channels. Along with this, there are some calcium channels which are voltage sensitive calcium channels. And what alcohol do is they inhibit glutamate release. So they will inhibit the release of glutamate into the synapse. Alcohol may also have some direct or indirect effects on reducing the actions of glutamate at postsynaptic NMDA receptors and at postsynaptic mGlutamate receptors. M means metabotropic glutamate receptors. So what happens? There is also an NMDA receptor here, and there is also postsynaptic metabotropic glutamate receptors here. So alcohol also have a direct or indirect effects on reducing the actions of glutamate at postsynaptic NMDA receptors and at postsynaptic metabotropic glutamate receptors. Alcohol's reinforcing effects are theoretically mediated not only by its effect on the GABA and glutamate synapses, but also by actions at opiate synapses within mesolimbic reward circuitry. Now we have already learned about this mesolimbic reward circuitry in the cocaine addiction and intoxication, but we will talk about this in more here because it is important to understand how the addiction develops and how the person gets the feeling of good and wants to continue to repeat those actions in order to get the euphoria, feeling good sense. This is the reward pathway. We are going to focus mainly on alcohol. Last time we focused on opioids, which is produced by the arcute nucleus, although even in alcohol it plays a role. So what happens, this is the ventral tegmental area of the brain, and it has got neurons which are sending axons to the nucleus accumbens, which is the main nucleus in the brain which is concerned with the reward system. If the dopamine is released here, the nucleus accumbens gets the reward, has the positive feeling, and wants to repeat that action again and again to keep on getting some dopamine here. Now, how is this dopamine being affected by alcohol? Let's take one thing at a time. This is the neuron in the prefrontal cortex, PFC, and these are glutamate neurons. They are in the prefrontal cortex. These are the one which mainly decides with the actions, planning, and they are excitatory neurons. Now they send connections to the ventral tegmental area, and within the ventral tegmental area they connect with dopamine neurons in the ventral tegmental area, and when they synapse with these dopamine neuron, they have certain receptors on their presynaptic neuron, which is glutamate neuron. Now they have a mu neuron, which is opioid receptor. They have this VSCC, which is voltage sensitive calcium channel. They also have GABA receptor. They also have this enkephalin, which is endogenous opioid, and they also have this metabotropic glutamate receptor, both at the presynaptic and the postsynaptic membrane in the synapse. And this glutamate is obviously released from the glutamate neuron into the synapse with the dopamine neuron. Now, how does alcohol play a role? Now, there is also another one, GABA-A receptor and GABA-B receptors. So what happens when we drink alcohol? It either, so opioid neurons, these are opioid neurons in the arcute nucleus, and they produce enkephalin. So this enkephalin also plays a role here. In opioid neurons synapse in the ventral tegmental area with GABAergic. So this is with the GABAergic. So within the ventral tegmental area, this is arcute nucleus. It's supplying to the dopamine neurons directly and also indirectly through this interneuron, which is called GABAergic interneuron. Now, when the opiate is released here, the alcohol occupies the mu channel, mu receptor, rather than this enkephalin, which is endogenous opioid, would have attached here and caused the effect to release more dopamine here. Here, the alcohol sits in that place with presynaptic nerve terminals of glutamate neurons. Inhibitory actions of opioids at mu receptors there cause disinhibition of dopamine release in the nucleus accumbens. Alcohol either directly acts upon mu receptors here or cause release of endogenous opioids such as enkephalin. So they do the same thing. They either make this release the enkephalin or they actually sit here and cause the same effect of increasing the dopamine in the nucleus accumbens here. Alcohol also acts at presynaptic, metabotropic, glutamate receptor, and voltage sensitive calcium channel to inhibit glutamate release. So this is here glutamate, this is the glutamate receptor from the prefrontal cortex. What happens is alcohol acts at the metabotropic. This is the metabotropic mGlutamate receptor. It acts here and also acts on the calcium channels, presynaptic calcium channels to inhibit glutamate release. So there is less glutamate release if the person is drinking alcohol. Alcohol enhances GABA release. So what we are saying is alcohol is releasing the GABA release and through direct or indirect actions at GABA-A receptors. So that's how ultimately what alcohol is doing here is it is affecting this production of dopamine at the nucleus accumbens. If it is more of dopamine produced here, which happens when the person drinks alcohol, they get the reward. They want to repeat the mechanism or repeat the behavior and the habit because they are feeling good, because the dopamine is being released. And if they don't, then they won't. So that's what it is, either through the glutamate receptors or through the opioid receptors or even through the intermediate actions of the interneuron GABA neurons. Opioid neurons arise in the arcute nucleus and project to the ventral tegmental area, synapsing on both glutamate and GABA neurons. The net result of alcohol actions on opioid synapses is thought to be the release of dopamine in the nucleus accumbens. Alcohol may do this either by directly acting upon mu opioid receptors or by releasing endogenous opioids such as enkephalin. These actions of alcohol create the rationale for blocking mu receptors with antagonists such as naltrexone. Presynaptic cannabinoid receptors of both glutamate and GABA synapses where alcohol may also have actions. This is the same one but created in a slightly bigger scale. So if you have missed anything in the first one, it will be more clear here. So these are coming from the arcute nucleus to the interneuron GABA neurons, and this is the GABA-A receptor, and this is the mu receptor here, and this is the GABA-B receptors. So they are ultimately affecting the nucleus accumbens to get more dopamine or less dopamine. So this is the most important reward nucleus in the brain. Cannabinoids, cannabis, or anything related with cannabis products like marijuana, hashish, cannabinoid antagonists such as rimonabant, which blocks cannabinoid 1 receptors, can reduce alcohol consumption and reduce the craving. Naltrexone blocks mu receptors and this blocks enjoyment of heavy drinking. Acamprosate is a derivative of amino acid taurine and interacts with both the glutamate system to inhibit and with the GABA system to enhance it, a bit like a form of artificial alcohol. So we have learned this is what alcohol does in the brain, and therefore acamprosate is doing the similar thing. Therefore we are calling it artificial alcohol. Too much glutamate can cause neuronal damage because if it keeps on firing, they are stimulatory and it goes in like in a chain reaction, firing, and that can damage the neuron. Disulfiram is another anti-abuse or to stop the alcohol, is irreversible, that means it is not reversible, inhibitor of aldehyde dehydrogenase. What happens when we drink alcohol? It is converted into aldehyde and then it is converted into acetic acid and then it is converted into acetyl coenzyme A, which is taken into the citric acid cycle to break down further and make energy out of it, producing carbon dioxide and water. If we inhibit this aldehyde dehydrogenase, what happens? Aldehyde will build up in the body, in the blood, and that buildup is toxic. This creates an aversive experience with flushing, nausea, vomiting, and hypotension, hopefully conditioning the patient to a negative rather than positive response to drinking. So every time a person drinks, they will get a buildup of acetaldehyde, which can cause nausea, sickness, and flushing, and even hypotension. So having a bad reaction. Other agents like topiramate, which is a 5-hydroxytryptamine or serotonin antagonist, and cannabinoid CB1 receptor, which we discussed earlier, may also help with alcohol problems. If you like this video, press the like button, share with your friends, and subscribe the channel. Thank you for listening, and I see you in my other videos.